Okay, are we putting the slides up, or do I do it from my, do I have control?
Tanya, you should be able to share the slides from your computer.
Right.
Your host.
You have control.
I'm just going to do that. So thank you, David, for the introduction. Okay, this should be good. So in less than 20 minutes, I'm going to try and give you a tour of the current state of drug discovery for tuberculosis. And as David has already given a great introduction to Mycobacterium tuberculosis, the one thing I wanted to sort of reinforce from the perspective of drug discovery is the complication of the disease itself. So as we can see, MTB can infect many different cell types. So we have macrophages, dendritic cells, we have free TB, and we have this very complex granuloma, very complex cellular structure. And from the drug discovery perspective, that poses particular challenges because we have to get drugs to the site of infection. Beyond that, we often think of TB as a respiratory disease. So yes, it does infect the lungs, but MTB can also infect pretty much every organ in the body. So we can have TB meningitis, we can have infection in the bones, we can have miliary. So again, when we're thinking about treating TB with drugs, it's pretty complicated because we have to be able to get to all of these different sites. And then the last thing that makes treatment of TB complex is the organism itself. So David mentioned this sort of cording and clumping, which makes drug access to the bacteria difficult. We have this very complicated and thick waxy cell wall, meaning drugs struggle to get into the cell. We also have, from the sort of practical perspective of doing drug discovery, the slow growing nature of the organism. It takes many weeks and months to do experiments. So drug discovery is a very long process. For those of you who are not familiar with the drug discovery and development process, here is the sort of outline of where we start. We start with screening and we end up with clinical trials. Now, this sort of pipeline makes it look very simple and straightforward, but one of the big things that happens during drug discovery and development is what we call attrition. So we have to start with millions of compounds at the beginning in our screens to end up with, one or two molecules that make it into clinical trials. And at every stage of this process, so every point in this process, we have massive attrition. Probably 80 to 90% of the projects fail at these different stages. So it is a challenging process. Despite that, if we look at the new TB drug pipeline in 2024, this is from the Working Group on New Drugs and is updated annually, we can actually see that it looks the most healthy it has for a long time. Now, although it looks like there are a lot of agents in here, two things you can see is, one is, if you look at the color coding, everything in the same color is the same class of antibiotics. So from that perspective, you're only ever going to have one of those make it into the clinic. So you can see already that there's not as many novel classes as appears at first glance. But also, as I mentioned, we have huge attrition during the process. So many of these projects will fail along the way at each stage. So I would say, although this is the healthiest we've seen it in certainly my career, there's still room for additional agents and more investment in order to get the drugs that we need to treat the issue. Again, if you go to the Working Group on New Drugs site, you can look at what's in drug discovery. So these are the projects that are prior to going into clinical phases. Again, it looks like if we look on the left, we have hit to lead, early projects. Lead optimization is later projects. It looks like we have a lot of molecules here, but again, most of these are going to fail at some point before they go forward. So we actually do need more. The very nice thing about this, though, is what we can see is there's a plethora of different organizations involved. So everything from here, we have academic institutions, we have large pharma, we have collaborations. It's very nice that we have all of these different organizations often working together to progress agents. We also have a very small, if we look the snapshot today, a very small group of agents that are in preclinical. So this is non-good laboratory practice state. Now, I mentioned that there's a huge attrition. If you look on the working group website, you will also see this list. And what I want to point out, very small at the top, it says, are these advancing? And the answer is no. So here's a list of many agents that in the last, few years have been worked on, but are not going forward. So you can see there's a lot of, you can see in real time this attrition. So coming back to the drug discovery process, which is what I'm going to focus on, I want to lead you to how we do drug discovery. So as I mentioned, we often start with screening in the field. This takes the form of screening these small molecule libraries. These can be anywhere from a few thousand up to millions of compounds. So we'll screen those. One of the challenges of MTB, as I mentioned, with the complexity of the disease, is we need to find new ways to screen. We need screens that are representative of infection. So there has been a lot of activity in terms of screening libraries, and the sort of simplest screens that you can do have been done and have interrogated all of the sort of commercially available libraries. So the focus now is really, can we develop novel screens that are going to look at different aspects of disease? And also, can we develop screens that identify agents that not only can be new drugs, but can lead to treatment shortening? So we can go from 6 months treatment down to 2 or 3 months. And one of the key aspects of when we're developing screens, as you can see a picture of our sort of liquid handling system, is it has to be adaptable to high throughput. So if we want to screen 2 million compounds, we really need to be running in 384, 5036 plates. So that can sometimes narrow the types of screens that you can do if you can't adapt them to this very high throughput. So what are the types of screens that have been developed in the field and within my lab as well? So what we will, as I mentioned, we need to be able to screen all of these, large libraries. So we'll develop high throughput screens. So here are some examples. This is just an example of where we can make the bacteria fluorescent, so we can easily measure growth. Or we can do an aerobic screen. So just looking at TB in culture. We can do intracellular, so inside a macrophage. We can make the bacteria have reporters, so they could report on, for example, cell wall damage or respiration. And then we'll screen, we'll use, plate readers, and then we'll generate a list of hits. Now, from a screen, this is the sort of data that we get. This is an example of a screen that we ran where we had about 200,000 compounds, so medium-sized screen. We ran it at a single concentration and we identified our hits. We got about 3,000 what we call hits, things that we thought were active. That's about a 1.5% hit rate, which is the average. From there, we need to confirm that they are actually hits, and we then determine the minimum inhibitory concentration by dose response. From this, we're able to then get down to about 1,000 compounds where the MIC was less than 20 micromolar. And you can see here, this is what our sort of distribution of MICs look like. We have many that are below this sort of cutoff of 20 micromolar. Some are very active, they are sub-micromolar. So that's an example of a standard screen against replicating bacteria. You end up with your 1,000 compounds you need to move forward with. And this is what a high throughput screen looks like when you're running single point. This is a screen where we were actually measuring the pH inside the bacteria and looking for molecules that would disrupt their ability to maintain their pH. And you can see here, most of the compounds don't do anything. The bacteria have a pH of 7, but we do see molecules that reduce the pH down to 6, and that's actually going to kill the bacteria. So that's an example of a type of novel screen that we can do. So once we have our screens, we have 1,000 hits at least, from a 200,000 library. The question is, what do we do next? And this is where we go into hit assessment. We obviously can't work on 1,000 compounds. We can't work on hundreds of different series. So we want to very quickly determine which are the molecules that are sort of the best to work on. And we do this by making, this is where chemistry comes in. We do this by making a small number of analogs, 30 to 50, with the goal of completing within 6 months, and then determining which of those series are the most attractive and tractable to move forward into the next stage, which is our hit to lead. In hit to lead, this is now where we're going to put a lot more effort into a series. We'll work on that, maybe only 1 or 2 at a time. We're going to make, several hundred analogs in a year to really explore the series and determine whether we can combine the anti-TB properties with drug-like properties. And the way we do this is when we're in hit to lead, we're not just doing an MIC. We'll be making compounds, we'll be doing the primary assay, doing the MIC, but then we layer on top the assays that enable us to look at drug-like properties. So now we're going to look at cytotoxicity, can we get selectivity for bacteria over human? We're going to look at what we call in vitro ADME. These are the drug-like properties. It's whether the molecules can get on board, whether they're absorbed, whether they're metabolized, how quickly they're excreted. We'll then look at in vivo pharmacokinetics, which is really a measure of if you treat with this agent, how does it get into the bloodstream? How long does it, will it last in the body? Can it get to the site of infection? And then alongside that, we'll be doing lots of other sort of microbiology to look at the properties. Can we attack non-replicating bacteria? Can we attack bacteria in the granuloma or in the caseum? Can we attack the intracellular bacteria, and can we attack bacteria in different metabolic states? So this is why we can't work on every series at this stage, because there's a lot of effort going into this. So from this, we'll get our compound activity profile, and then we'll go to an in vivo efficacy using the mouse model. So at this stage, we do have criteria on how do we determine which series to move forward. So for every assay we run, we have a criterion, for example, with MICs, we want them to be less than 5 micromolar, and we want them to be at least 10-fold selective for the bacteria. We want to make sure that they work for the lineage strains that David mentioned. We don't want, any strains to be resistant. And then when we go to this in vivo mouse model, we want to see that they work and that they can give us a reduction in the lung bacterial burden. And then one of the other things that we're very focused on is what we call, this is HERG. This is a measure of cardiac toxicity. And again, this is a very important one for us that we're actually including this. We want selectivity so that we're not going to have any cardiac issues. So once we have a series that's gone through hit to lead, our next stage is then to go to what we call lead optimization. In lead optimization, we're really now changing the focus. In hit to lead, we're really exploring the series and making lots of very different analogs and testing the drug-like properties. In lead optimization, we have an idea of what the final molecule is going to look like. We'll explore around that molecule, but we're also now a bit more focused on safety and the drug-like properties as opposed to the antibacterial properties. And now we're going to be making fewer analogs, but it's going to take a little bit longer because of the types of assays we do. So again, when you go to lead optimization, you're going to have a lot of different assays. This is what we focus on. We want to remove any remaining liabilities, focus on safety, the in vivo activity, but all the while maintaining the antibacterial potency and the drug-like properties. Again, we have a set of criteria. I won't go through them, but we have this long list of criterion that molecules have to meet. And you can see now why at each stage we lose a lot of molecules. They might have very good antibacterial activity, but they fall short in all of these other areas like safety. So I want to wrap up by giving you an example of drug discovery and action of how do we get from our molecule through at least to in vivo proof of concept. So one of the things that came out very early in the aerobic screening front run by many different groups was we all were finding these very different structure molecules. You can see here there's not any structural similarity in these molecules, but they all appeared to hit the same target, and that target was MMPL3. And it turns out that targeting the cell wall is very effective because obviously the cell wall is this barrier, but this is the part of the cell wall that we found that everybody was hitting. So here we have the inner membrane and the outer membrane, not to scale, but what the target we were hitting is this protein MMPL3, which sits inside the inner membrane, but it's actually exporting the components of the outer membrane. It's exporting this molecule here, TDM. And it turns out that this is a very common target. I mean, I showed you 6 or 7 scaffolds. There have been, 10, 20 scaffolds that actually hit the same target. And it appears to be what we call very vulnerable. We also knew at this point that MMPL3, at least from the genetic perspective, is a vulnerable target. Via work done in Dirk Schnappinger's lab, this is looking again at an in vivo model, and this is actually switching off production of MMPL3 at different times during infection. So here we see this is what normally happens is when you infect mice, you get an increase in the bacterial load in the lung over time. But if you now at point here or point here in red, if you switch off the production of MMPL3, you actually get clearance of the bacteria. Not only do they not grow further, they're actually cleared from the body down to here, the limit of detection. So this was very nice confirmation that we knew that if we hit this target, we should be able to clear the bacteria from, at least from the in vivo model in the mouse. So from this, our own group decided that we would develop at least one series of MMPL3 inhibitors, and this is just an example of, here's the structure. In our process, what we do is we divide the molecule into these 3 areas. We play around with like different analogs. We change all of those pieces for different pieces, and then we get our best molecules. And here's an example of one of our better molecules where we have, this is looking at in vitro killing of the bacteria. So now we're just measuring viable bacteria. If we treat with this agent, we can see that we kill the bacteria quite quickly. We do see the appearance of resistant mutants after some time, but that's because we have quite a lot of bacteria at the beginning. So this is very nice in vitro. The final step was to say, okay, we think this should work in vivo. So what happens in the mouse model? So this is confirmation in the mouse model, the acute mouse model. Again, we're just measuring the bacterial burden in the lung. This is at the start of treatment. This is untreated, so you can see the bacteria is still growing. When we treat with our molecule, we do see that there isn't as big an increase in the bacteria. So we are having an effect on the bacterial load. So this again in vivo is a sort of confirmation that these molecules can move forward. And these molecules are now what we call, they're still in the late lead optimization stage, ready to go forward to sort of safety testing. So the last thing I wanted to mention about how we do drug discovery is it takes a village. There are many different disciplines required. We need microbiology, chemistry, pharmacology, toxicology, and all of those expertises are very rarely found in one organization. So one of the large consortia that we're part of is a TB drug accelerator. And you can see there's more than 20 sort of participants in this. And there are many other large consortia like this globally who are really working together to do drug discovery. Finally, I'd just like to acknowledge our funding. This is the group in my lab who do a lot of the work that we work on. And again, one final plug for the C-Track Basic Science Core. For any of you who are interested in working on TB but don't know where to start, we provide a consultation service that you can access. And with that, I think I will wrap up, hopefully on time.